Let's take a look at the rather complicated life cycle of the red blood cell. This particular document that you're looking at can be found in D2L in the extra notes and resources, and it will be under the docs. Okay, so if you want to print that off and have this in front of you while you're going through this Explain Everything, I am going to compare the flow chart that I have here to the actual diagram. Let's compare the flow chart that you have in front of you with the diagram. So let's take a look here and start with number 1, which is the red blood cells being broken down into their component parts by phagocytosis by macrophages in the spleen, liver, or bone marrow. And let's just take a look back now at the flow chart, and this will obviously be right here, and this will be number 1. Now let's look at step number 2, which is the components of the red blood cell being broken down into its component parts, which is going to include globin, which is going to be the protein part, and the non-protein part, which is going to be the heme. Looking at the flow chart, we can see that this is occurring right here, where it is being broken down into globin and into heme, so that would be step 2. After the red blood cell has been broken down into its component parts, there is then going to be 2 different paths that the components are going to take, and this is going to be number 3 right here. Okay, the globin, the protein, is going to be broken down into its component amino acids and then be reused for protein synthesis. Okay. The non-protein part, the heme, okay, number 4 here, is going to be broken down, and the iron will be released from the heme. Go back to our flow chart. Steps 3 and steps 4 are right here. Here is the globin being broken down into amino acids and reused for protein synthesis, and here is the heme, number 4, being broken down into iron. Let's follow the path that the iron is going to take. So number 5 is that it is going to be transported, transferred in the blood via transferrin. Okay, so transport transferrin. Looking back here, number 5 is going to be right here where it, where it combines with transferrin for transport in the blood. The iron combined with the transferrin is then going to be transported to the liver or muscle, and then it is going to combine with ferritin. So let's just note here that it was in the blood being transported via transferrin. Once it gets into the liver or the muscle, it is combining with ferritin, and the purpose of this is for storage. Okay, and remembering why we're talking about transferrin and ferritin is because the Fe3+ ion is called the ferric ion. Okay, continuing to look at the flow chart now, we will see that the transport of the ferric ion combining with transferrin, going to the muscle and the liver, and then being stored is going to be number 5, and it is right there. When needed, the iron, the ferric ion, is going to be released from the liver, and of course it's going back into the blood, so it is going to have to recombine with transferrin to be transported in the blood. Okay, and number 6 right here, okay, released from storage and is going to combine with transferrin. Once again, transferrin for transport in the blood. The transferrin is then going to transport the iron back to the red bone marrow, number 7 here, and in the red bone marrow the iron and the globin will recombine to form the hemoglobin, and the red blood cells will be reproduced by the process of erythropoiesis. Number 8. Okay, looking here, we have number 7, which is the transport back to the red bone marrow, and then erythropoiesis, and the hemoglobin synthesis is number 8. Let's go back now to the original steps where we were looking at what happened after the phagocytosis of the red blood cell. we looked at the fact that the protein part is going to get broken down into amino acids and then reused for protein synthesis, and that the heme component was broken down, okay, and the ferric ion followed the path that we just took. Now what we have to look at is what happens to the non-iron component of the heme group. And so now we're going to be skipping to stage 9. Okay, there's another little step there, but we're not going to talk about it too much. Let's just say that overall the heme is converted into bilirubin. Okay, so that's step number 9. And on our chart here, we're going to see right here that the non-iron component of the heme, okay, is getting converted into bilirubin. The bilirubin then is going to go to the liver, as you can see here in step 10, going to the liver, and then will be released in bile, and that's why this little arrow here is green. Okay, it will be released in the form of bile, okay, step number 11, into the small intestine. So going back to our flow chart here, we have step number 10, which is it going to the liver. And then number 11 is it's being released in the form of bile into the small intestine. Following the path, we'll see that the bilirubin is then going to be converted via bacteria into something called urobilinogen. Okay, that is going to be number 12. And then via the blood, it will be transported to the kidneys, where it will be converted into urobilin or urobilin. Okay, and this is the component that causes the urine to be a yellowish color. So number 12 and 13, if we look at the flow chart right here, we will see that number 12, via the bacteria, the bilirubin is going to be converted into urobilinogen, okay, then going to the kidneys to be converted into urobilin and secreted in the urine. Going back for a minute, looking at the bilirubin, you can see that there's an alternate pathway that the urobilinogen can take, and that is going to be number 14 here, where it is converted into stercobilin, okay, stercobilin, and it will be excreted in the feces. The stercobilin is the bile pigment that gives the brown color of human feces. Going back to our flow chart, the final number 14, right here, where the stercobilin is excreted in the feces. The final point I'd like to make is to note that this is a cycle. Okay, so there the red blood cells are being broken down, and then in the red bone marrow they are being resynthesized, and just note here that they will circulate in the blood for approximately 120 days. Let's put all the numbers on this one flow chart, starting with number 1, where the red blood cells are being broken down by phagocytosis in the spleen, liver, and red bone marrow. The number 2 is where the hemoglobin is going to be broken down into a protein component and a non-protein. The protein will further be broken down into amino acids and reused for protein synthesis, where the heme will be broken down into an iron, okay, a ferric ion, and combined with transferrin for transport into the blood to the muscles and the liver, and then be stored, and if needed will be released from storage, recombining with the transferrin in the blood, transported to the red bone marrow, and then resynthesized. The hemoglobin will be resynthesized, the red blood cells will be resynthesized via erythropoiesis, and then put back into circulation. Looking now at what happens to the non-iron component of the heme. Okay, number 9 is that it's going to be converted, okay, into bilirubin and then transported into the liver, okay, which is number 10, where it will be released in the form of bile and go to the small intestine. Okay, from the small intestine, it will then go into the large intestine and be converted by bacteria into urobilinogen, and then it has two possible fates. It can either be converted to stercobilin and excreted in the feces, or it can go to the blood and be released from the kidneys in the form of urobilin. So this was number 13, and this was number 14.